In our last lecture, we finished with the structure of proteins, particularly looking at the basic units of proteins, amino acids, and we looked at some other properties of amino acids, classifying them depending on their side chains. And then we even looked at the various levels of organization of proteins, the primary structure of proteins, secondary structure, tertiary and quaternary structure. And I told you that because this course is particularly on metabolism, we will take the specific function of proteins as enzymes, because we will meet enzymes throughout, so that you will be abreast with the functions of enzymes. Okay, our class rep is now writing me, letting me know that you have just closed. All right, so we continue. So what are enzymes? First of all, let's get to know what enzymes are. The reaction which you are seeing on your screen is a common reaction that you refer to in metabolism. So this is sucrose that is undergoing metabolism. So you have sucrose is sugar, common sugar. That's the sugar we use, the table sugar. So it's getting oxidized, and in that process of oxidation, it gets broken down into carbon dioxide, which we say carbon dioxide is a byproduct of metabolism. Some amount of water is generated in that process, and then energy is given. So that is what we say, that carbohydrates are broken down to give energy, and it gives out carbon dioxide and water as byproducts. Now, when you take this reaction and you are applying it, practically, as you know, I just said this is sugar. If you put sugar on your table in the kitchen or your dining table, you don't go and come back and see that the sugar has turned into carbon dioxide and water. It's like the sugar is no longer there. It's turned into carbon dioxide gas, and then maybe some water inside the bottle. You don't see that happening. The sugar will be there. Maybe because sugar is hygroscopic, it can absorb water from the atmosphere if you put it down for a long time. If it's a lot in the jar, you can see it's gotten kicked. Even if you have the sugar very small at the bottom, because it absorbs water, sometimes maybe it may become sort of liquid in a way, but it is still the sugar there. If you taste it, it's still the sugar. It's not like it has turned into water. And yet we are saying that sugar breaks down into carbon dioxide and water when you take it into your system. How does that occur? So we have what we refer to as enzymes, which are mostly proteins that are responsible for catalyzing biochemical reactions. I mean, to speed up the rate of the reaction. These are biocatalysts. And most of them are proteins. Most of these enzymes are proteins. A few of enzymes are RNAs. And those RNAs, we refer to them as ribozymes. And they are mostly involved in metabolism of nucleic acids. So the recycling of nucleic acids, breaking down DNA, RNA molecules, to go back into the synthesis of these nucleic acids again. So we'll see that actually it is because of the action of enzymes that you can have such a reaction occurring very fast in the body. The moment you take in sugar, within, a couple of seconds, that the sugar gets into your bloodstream, gets to the cells, the sugar is quickly broken down and it gives you energy because of the action of enzymes. So enzymes are needed for the sustenance of life because the reactions that are occurring in us, they need to occur at a fast rate and be coordinated. It can be slow. If it's slow, it means our life cannot be sustained. So yes, this reaction actually, as I was saying, that if you put sugar there, you will see that the sugar is still there. It's not that the reaction is not occurring. It is actually occurring, but it's occurring at a very slow pace that is negligible. One thing you must know is that an enzyme will not come to cause a reaction. No, the enzyme comes to speed up the reaction that is already occurring. So if a reaction will not be occurring at all, an enzyme will not come and make that reaction occur. The reaction must already be occurring spontaneously before the enzyme will come or a catalyst will come to speed up the reaction. That is basic principle of the action of catalysts. So for instance, if you were in a lab and you are running some physical reaction in the lab, one of the things that you do to speed up the rate of the reaction is to apply heat, that is to give energy to the reactant molecules, and then the reaction will run fast. But that's not an option for biological systems, because we learned biological systems are made up of biomolecules. And some of these biomolecules that we refer to, like the proteins and nucleic acids, if you increase temperature above the physiological temperature, I mean 36.6 degrees, approximately 37 degrees Celsius, by the time you get to 39, 40, you start having what we refer to as denaturing. The system will break down. So we cannot have increase in temperature as a way of increasing the rate of these biochemical reactions. So this is where the enzymes, come in to do that. So before we continue, let's take a look at a basic principle of the activation energy profiles of reaction. It's a basic principle by which all reactions occur, and that is that all reactions that are occurring in between molecules, or say any chemical entity that is reacting at any point in time, you must know that that chemical entity exists because it has a certain stable energy state. So that if you say sucrose or you say glucose, glucose is existing at a certain stable energy state. If that stable energy state was not there, then that compound will not be existing in the first place, because You see, wherever you are now, wherever you are sitting, wherever you are watching this or listening, at any point in time, you are seeking a comfortable position. If you are not comfortable at any moment in time, you will adjust yourself. And if you are already comfortable, then you just remain where you are. There's no need for you to change your position. So it's the same thing at the molecular level. That if you take reactants, so what you see on the left-hand side is a reaction, it's an energy profile of a reaction that if you have reactants on the vertical axis, we are seeing energy, and on the horizontal axis, just reaction coordinates, so that you have reactants at a certain energy level. They are at a stable energy level already. There will be no point for them to react to form something else, a product, because they are already stable at that state. In order for them to react, they must be at a certain energy state that they will be unstable. And so for a reaction to occur between reactants, there must be what is referred to as activation energy. So the energy level of the reactants is raised up by activation energy, where they are at a transition state. At that state, they are unstable. They are most unstable, that they begin to look for a new stable energy state. That is when the reactants will react between themselves and form a product that will have a lower energy state, which is even lower than the energy state that the reactants were occupying. Now, the blue curve represents the energy profile of a non-catalyzed reaction, a reaction that is not having any catalyst there to speed its rate up. Because we are talking particularly about enzymes, this blue line is being referred to as the energy profile of a non-enzymatic reaction, because the enzyme is the catalyst. So it's a non-enzymatic reaction. Now, you see the orange line represents the energy profile of an enzymatic reaction. That's a catalyzed reaction. And you quickly see that from the energy level of reactants to the energy level of the transition state for that enzymatic reaction, which is the activation energy of that enzymatic reaction. That activation energy is lower than that of the non-enzymatic reaction. So that is basically how enzymes are able to speed up chemical reactions. Because, logically, what it means is that a reaction which is not catalyzed will take a longer time for it to reach a transition state for the reactants to react, because it requires more energy. But if you see a reaction, the orange line that is having a lower transition state, what it means is that it requires a lesser amount of energy to reach a transition state, so that the reactants will react between themselves and form a product. So that can occur in a shorter time. You even see what happens first of all is that an enzyme binds with a substrate, that is the substance on which it is acting, and that gives you an enzyme substrate complex. What is written as ES complex is referring to an enzyme substrate complex, which is having an energy a little lower than that of the reactants. And then you have the activation energy that takes that enzyme substrate complex to a transition state, and the product is formed. The product which is formed in the two reactions, catalyzed and non-catalyzed reaction, is the same. The only thing is that the rate of reaction of the catalyzed reaction is faster. The reaction proceeds at a faster rate than that of the non-enzymatic reaction. All right, so that understood. Before we continue talking more about enzymes, there are some simple terminologies that we should get abreast with, because it's language. Every field has its own language. Accountants will have their language. Marketers will have their language. You are more or less chemists, biologists, and specifically we are now talking about enzymes. So it's like some fundamentals of enzymology. You have some terminologies that are used that when we continue and we mention some of these things, we should quickly relate and understand what the conversation is about. The first is cofactor. A cofactor, you see, I mentioned that most enzymes are proteins. A cofactor is an inorganic non-protein part of an enzyme. It's non-protein, sometimes being referred to as a prosthetic part of the protein. It's required for the protein to be active as an enzyme. If it's not there, the protein will not be active. That's the cofactor. You come to see the cofactor. The cofactors are inorganic metal ions. When we continue, we'll see that. Then we see coenzyme. Sometimes when you read biochemistry books, cofactor, coenzyme are used interchangeably, but there is distinction between them. Coenzymes are organic non-protein parts. They are also non-protein parts of the enzyme, but they are organic. And we'll also see soon that the coenzymes are mostly some derivatives of vitamin Bs. Then we see apoenzyme. So apoenzyme refers to the protein part of the enzyme. That's the protein itself. That's either a cofactor or coenzyme is binding to make it active. Active site. Now, an enzyme is a protein, a big protein. It doesn't mean that at any part of it at all, a reaction will be occurring. There's some specific part of an enzyme, that protein structure, that the reaction that it is catalyzing will be occurring. Usually, you have active sites made up of 2, 3 amino acids. As the protein folds, they form something like a pocket. An active site can be just one amino acid anyway for some proteins. And it is at those particular positions that reactions occur. And we have activation. So any process that will sort of increase the activity of an enzyme or initiate the activity of an enzyme, that process is activation. And the opposite of that is inhibition. So inhibition will either slow down the activity of an enzyme or terminate the activity of the enzyme, stop it altogether. That's inhibition. And any compound or any chemical entity that does that carries out inhibition is an inhibitor. The same way for activation. Any chemical entity that will initiate or increase the activity of an enzyme is an activator. So for instance, there are some of these metal ions that we refer to. They are activators of the enzyme. And we have substrate. So the substrate is the compound on which the enzyme is acting. So for instance, that reaction we saw from the beginning, where we saw sucrose, that was getting broken down into carbon dioxide and water and giving out energy. The substrate there is sucrose, the sugar. So an enzyme that is acting on that sugar is a substrate to the enzyme. All right, so these things understood, we can move on to how enzymes are named, naming of enzymes, because we'll be seeing some enzymes and you see some names, and you should be able to see what the enzyme does just by looking at the name of the enzyme. When you just see the name of the enzyme, you should be able to know that this enzyme will do this. One of the ways that enzymes are named is just by adding the ending, this suffix ase, A-S-E, to the name of the substrate on which the enzyme is acting, the compound on which the enzyme is acting. You just add the suffix ase. So for instance, the first example you see here is urease. So the suffix is there, ase. But this is acting on urea. The substrate is urea. So what the enzyme does is it catalyzes the breakdown of urea, hydrolysis of urea. That's what urease does. Lactase. So lactase also acts on milk sugar lactose. So it's also catalyzing the metabolism of lactose, break down lactose into glucose and galactose. So just by looking at the name of the enzyme, you can easily tell that this enzyme will act on this substrate. Another way that enzymes are named is based on the type of reaction that the enzyme is catalyzing. So when you hear a name like DNA polymerase, what the enzyme is doing is catalyzing the process of polymerization of nucleotides. So nucleotides are the monomers of nucleic acids. And bringing the nucleotides together, meaning that you are forming a polymer, the nucleic acid, which is DNA that you are forming, is a polymer. So you are having the substrate is DNA, like, it's a formation of DNA from nucleotides. I see the name is polymerase, and that is based on the process polymerization. So you see, it is still ase that is being attached, but this time around the ase, that suffix ase, A-S-E, is added to the process, not to the name of the substrate, but to the process. The next example you are seeing is pyruvate decarboxylase. Decarboxylase. So again, yes, the substrate is pyruvate, and the process on it is removal of carboxyl group, the carboxylic acid group, COOH, the removal of that group. So carboxylase. So you can take it as deleting. So deleting of, so that is removal of carboxyl group from pyruvate. Then we have the third one. This one, the ending is zyme, just like the enzyme, the name enzyme, zyme. It's a lysozyme. and it's also based on the process lysis, which is the breakdown. So breakdown of what? Bacterial walls. So lysozyme is involved in the breakdown of bacterial walls. All right. But there can also be some common names which do not follow any of the systems that we have looked at. So for instance, pepsin. Pepsin is a common name. You can see pepsin, and in the name, you will see what it does. Because it's one of the first enzymes that were discovered, studied, separated from digestive juice. And because it was separated from digestive juice, it was the name, was linked to digestion. And the Greek word for digestion is pepsis. So it has that Greek root. The name has a Greek root, pepsis, meaning digestion. Then you have another one, trypsin. Trypsin too is a common name, also one of the first enzymes, to be extracted. This time around, it was extracted just by rubbing glycerol on pancreatic tissue. So on the pancreas was rubbing some glycerol on it to extract the protein, the protein which is trypsin, into the glycerol. And because of that process, which is to wear down something, to scrap off something, the word for it in Greek is trying. So from trying, you have the name trypsin. All right, so but these are common names, so you can't just see the name and then you'll be able to tell. Now we can continue to look at classification of enzymes. Enzymes can be classified into 6 major groups. And the first group is what we refer to as oxidoreductases. So we are looking at classification of these enzymes based on, again, the type of reactions that they catalyze. And the first one is oxidoreductases. These catalyze oxidation and reduction reactions. You know the reactions you commonly refer to as redox reactions. These enzymes catalyze those reactions. An example you are seeing is lactate dehydrogenase. That's an enzyme which is an oxidoreductase. And what is it doing? It is here oxidizing lactate into pyruvate. So for instance, if you look at the structure of lactate in the middle, the carbon there is a hydroxyl group on it, OH. The product that is forming is pyruvate. And you see in place of that OH, that hydroxyl group, you see a double bond O. So the hydrogen is given out. That's an oxidation reaction occurring over there from lactate to pyruvate. And the enzyme that is doing that, lactate dehydrogenase. So lactate is the substrate, and so dehydrogenase. And you see, hydrogen is gone. And you are left with just oxygen over there. Actually, when you see the name of any enzyme with this word in it, dehydrogenase, quickly you must know that that is an oxidoreductase. In fact, the word dehydrogenase is also used sometimes in place of oxidoreductase. So for instance, when the classifications are being done, like in place of oxidoreductase, you can see that that first group will be dehydrogenases. because dehydrogenases are the same as oxidoreductases. All right, then the second group is transferases. And this is also simple for you to remember because of the word transfer. So these enzymes will be involved in transferring of functional groups. Let's say transferring of a group from one molecule to the other. And you see an example here, aspartate aminotransferase, which is also known as aspartate transaminase. So what does it do? I mean, then if you look at the two names, the first one says aminotransferase. So it tells you that it transfers amine groups. Then the second name also tells you that because it says transaminase. So it transfers amine group. Now, when you look at the example of the reaction in which this enzyme is acting, you see it's a common reaction we will see later when we are looking at metabolism of proteins. It's what we refer to as transamination, when the amine group in an amino acid is transferred onto a sugar. So you see alpha ketoglutarate is an intermediate carbohydrate in metabolism of carbohydrates. And you see there is an amine group on aspartate. That's an amino acid, aspartic acid. We saw the amino acid. So even if you forgot what you did in first year, you saw it again at the beginning of this course. So aspartate, the same as aspartic acid, you see there's that amine group there, NH3 plus. Now, if you pay attention, that NH3 plus will change a position with this C double bond O, which is on alpha ketoglutarate. So let's look at the product. The product, oxaloacetate. Oxaloacetate. Compare oxaloacetate to aspartate. And you will see that in place of the NH3 plus, that amine group in aspartate, you are now having C double bond O. And that C double bond O is coming from the alpha ketoglutarate. And if you also look at glutamate, compare glutamate to alpha ketoglutarate. And you will see that in place of the C double bond O in alpha ketoglutarate, you have NH3 plus. And that NH3 plus came from aspartic acid, NH3 plus. So it's just an exchange. There's an exchange between this C double bond O in alpha ketoglutarate and the NH3 plus in aspartate. There's that exchange between the two groups. And the enzyme which is involved in that process is aspartate transaminase or aspartate aminotransferase. So it's transferring the amine group from the amino acid onto that carbohydrate molecule. So you come to see that the processes of metabolism, when we are saying metabolism of proteins, it's not like it's in a vacuum, it's on its own. All the metabolic processes are interconnected. So this is where you see metabolism of protein connected with metabolism of carbohydrates. All right. Then the third group is hydrolysis. Hydrolysis. So hydrolysis will catalyze what we refer to as hydrolytic reactions or hydrolysis. And an example is acetylcholine esterase. Hydrolysis, you have in the presence of water, a molecule broken down into smaller molecules. So we see acetylcholine is broken down into acetic acid. So it's just this first part of that molecule gets broken down into acetic acid. Now this water molecule, take it out, it will give you an H plus and OH, which means that OH comes, let's say, the hydrogen from water comes to bind on that oxygen of the acetic acid residue there in acetylcholine to give you the full acetic acid. And then the hydrazole now comes to bind at the end here to now give you the choline molecule. So you have acetic acid and choline. This reaction is a hydrolytic reaction. It's hydrolysis, the breakdown of acetylcholine in the presence of water. And the enzyme which is doing that is acetylcholine esterase. All right. The fourth group is lyasis. Lyasis. So lyasis, we're looking at the example aconitis. When we are looking at the TCA cycle, the tricarboxylic acid cycle, what is also referred to as a Krebs cycle. I'm sure you did some Krebs cycle in first year, but you may not have paid attention to maybe some specific reactions. This time you take the reaction and you pay some attention to it. If you go back to that TCA cycle, you will see this reaction. See this reaction over there. And you see cis-aconitate forming isocitrate. Now, you see this reaction, there's water here. So you can easily confuse it with hydrolysis. But this is different. It's not the same as hydrolysis. You see, hydrolysis, there was one molecule with water, and that molecule was breaking down into two molecules. This time around, that's not what you see. You see that the water molecule actually, combines with this molecule. So you now see there are two reactants, but what you have here is one product. And you see there's a double bond in cis-aconitate. That double bond is no longer there, it's removed. How? Because you can see on this carbon in cis-aconitate, where you have CH with that double bond, you now see in isocitrate, you see a hydroxyl group on that carbon. So that hydroxyl group is coming from the water molecule. And you also see that the second carbon with a double bond that has COO minus on it in cis-aconitate, that carbon is now having hydrogen on it. So you see the water molecule is broken down. Hydrogen goes to this carbon, and then the hydroxyl group OH comes onto this carbon, and the double bond is removed. It's actually a reversible reaction. So you can either have that removal of the double bond, or if it is the reverse of the reaction, the formation of a double bond. And the enzymes that catalyze such reactions are referred to as lyases. The fifth class is isomerases. So coming from the word isomerization. So you can quickly understand that. I mean, well, what are isomers? Isomers are compounds with the same chemical formula, but different chemical structures. So isomerases will be involved in the formation of, say, the isomer of a substrate. So for instance, the example you are seeing is phosphohexose isomerase, which is acting on glucose 6-phosphate. This one too, you have seen it when you were looking at glycolysis in first year. So glucose 6-phosphate is converted to its isomer, fructose 6-phosphate. And the enzyme which is acting in that process is phosphohexose isomerase. It's an isomerase that is acting on the substrate phosphohexose. What's a phosphohexose? A phosphohexose is a 6-carbon sugar with a phosphate group. So you see, glucose 6-phosphate is a phosphohexose because it contains 6 carbons and there's a phosphate group there on carbon number 6. That's why it is glucose 6-phosphate. The same way you see fructose 6-phosphate is also a phosphohexose. It's an isomer. So same chemical formula, but different structures. See, the glucose 6-phosphate is having a 6-sided ring. That's a pyranose. Whilst you have the fructose 6-phosphate being a furanose, is having a 5-sided ring. Then the last one is ligases, also referred to as synthetases. So these catalyze the joining of 2 molecules, particularly you see DNA ligase, which is involved in the synthesis of DNA. Nucleic acids after the process of replication, that is the production of DNA from another DNA molecule. There will be some gaps that are left in between the some nucleotides at the ends, and after that you have DNA ligase coming to seal those gaps. Those gaps are sealed, and that's a common example of ligases. So that's what they do. They join molecules together. Okay. Now we can move on to look at classification of enzymes based on their specificity to the substrate on which they are acting. And these two, we can break that classification into 4 main groups of enzymes. And the first group of enzymes we will refer to as absolutely specific enzymes. Now, absolutely specific enzymes will act on only one substrate, just one. They will not act on any other. So for instance, lactase, the name we saw earlier, lactase. Imagine if lactase was going to act on another substrate apart from lactose, and the name is lactase. I mean, then the name will be misleading. But see, the name tells you that this enzyme is substrate specific. It acts only on lactose, nothing else. So it is absolutely specific to lactose. The same way you see the name glucokinase. This is an enzyme that is also absolutely specific to glucose. That's the only sugar it will act on. It does not act on any other, only glucose. Now you see the names of enzymes with kinase. If you see the name of any enzyme with kinase, it means that enzyme is involved in the transfer of phosphate group. Usually, they will transfer phosphate group from ATP, adenosine triphosphate, a cellular form of energy, onto a molecule to activate that molecule. It's a way of activating molecules to go down a certain reaction path or make them active to carry out a function. So kinases are enzymes that are involved in the transfer of phosphate groups. It can be phosphate groups from one molecule to the other. All right. The second is what we refer to as group-specific enzymes. So these ones, they will act on a group of substrates, not only one. So if you see hexokinase, see if you compare hexokinase to glucokinase, see glucokinase is specific to glucose, but hexokinase will be acting on all hexoses, on all 6 carbon sugars. So not only glucose, can act on fructose as well. Allose, maltose, all the 6 carbon sugars. Hexokinase can act. Pepsin. So pepsin will act on peptide bonds that amino acids with aromatic side chains form. So this one too is not only a single amino acid, because we saw the amino acids and you saw three amino acids that have aromatic side chains, which are phenylalanine, tyrosine, and tryptophan. So pepsin will recognize that amino acid, and then it goes in to break the peptide bond that amino acid has formed with other amino acids. Because the pepsin is actually involved in the breakdown of proteins that are referred to as less digestible proteins. Because they have these hydrophobic side chains, which means they are going to be less soluble. So they require sort of more energy to be broken down. They are more difficult to be broken down. And so that's where an enzyme like pepsin acts. I mean, similarly, you have trypsin that will also be acting on the peptide bonds that amino acids lysine and arginine form at their carboxylic acid end with other amino acids. Lysine and arginine are also similar. If you go and look at the structures of the amino acids that we saw, you realize that lysine and arginine, they have an extra amine group in their side chain, which is carrying a positive charge, a net positive charge on the amino acid molecule. So see these things, when you are studying them, you have to be going back to the amino acid structure and look at it. Then you can easily compare, like for instance, you compare lysine and arginine and see the similarity between them. Because now we are saying group specificity. So what is common between these two that trypsin is recognizing them to be acting on? The same way when we refer to aromatic side chains, like amino acids with aromatic side chains, if you forget, you just go look at the amino acid structures and you see phenylalanine, tyrosine, tryptophan. These 3, they are the amino acids with aromatic side chains. Then you see the similarity between them. When you refer to hexokinase, and we say they are acting on hexoses, which are 6 carbon sugars, you go back to first year and go and look at when you were looking at the structures of the carbohydrate hexoses. and you look at them again and remind yourself, because this biochemistry will be following you throughout. So if you threw your notes from first year away, go and look for it, because you will need it throughout. Even in your third year, you will need it. All right. Then the last one is enzymes that are acting on specific bonds. So these ones are specific, a particular bond that they will be acting on. And an example is alpha amylase. Alpha amylase. So you see, we have alpha amylase. The salivary amylase that we refer to is an alpha amylase. And that alpha amylase breaks down alpha 1,4-glycosidic bonds. These are the bonds that you find between glucose molecules in starch and the storage form of carbohydrates in mammals like us in the liver. So amylase can break down the glycosidic bond, which is between starch and glycogen, release energy to the body. But again, back to first year, you learn something about the Haworth perspective formulas. That is the ring structures of the monosaccharides. And you saw that on the anomeric carbon, the first carbon, carbon number 1 of the monosaccharides, you can have rotation of the groups. That is the hydrogen and the hydroxyl group on these. And when you have the hydroxyl group pointing downwards, you have an alpha configuration of the monosaccharide. If you have the hydroxyl group pointing upwards, you have the beta configuration. So you can have beta 1,4-glycosidic bond. That type of bond is found in cellulose, which is the main component of the woody part of plants. Let's say fiber. You see, when you eat, there's a part of the food which is indigestible, what you refer to as roughage. The reason it's indigestible is because your amylase cannot break down beta 1,4-glycosidic bonds in the fiber. And so it just comes out. So that is bond specificity of alpha amylase. Another interesting thing about it is, you see, for instance, you see termites eating wood. The termite itself is not benefiting from that wood because it also does not have the enzyme that can break down the beta 1,4-glycosidic bonds, except that you have the termite having some microorganism in its gut, its digestive system. So that's a trichonympha that has the enzyme cellulase, and cellulase can break down the beta 1,4-glycosidic bonds. That's why you see termites eating wood. All right, let's continue. The fourth is stereospecificity. Stereospecificity. So we have stereospecific enzymes or stereochemical specific enzymes. See, these ones act on a particular isomer of a substrate. So usually you have, you remember what you refer to as enantiomers. These are isomers of a compound that are mirror images of each other. They are non-superimposable. you refer to them. You saw these isomers, D and L isomers, particularly with amino acids. Now, when we go to look at metabolism of amino acids, there are some enzymes there that are specific to the L isomer of amino acids. And you have some that are specific to the D isomers of the amino acids. So we have the L amino acid oxidase and we have the D amino acid oxidase. These are specific to these isomers. Actually, in biological system, most of these biological system, most of the amino acids are in their L isomeric form. Actually, the L isomeric form is like the active form of the amino acids. So for instance, when we consume proteins, sometimes we do take in some D amino acids, particularly from plants. But you see, even in the metabolism of these amino acids, when there is D amino acid, that D amino acid gets converted to L amino acid before the metabolism process continues. So L and D and L amino acids, they act like some biological switches, like on and off. So if you have the L isomer of an amino acid, that amino acid is active and can carry out a particular function. But if it's the D isomer, then it is inactive. And so a particular reaction will not proceed. So that's how they act, on and off, like biological switches. All right. Then we saw coenzymes and cofactors. We refer to them as the prosthetic part of enzymes, the non-protein part of enzymes. So what you see being depicted on your screen is the first here you see is apoenzyme. That is the protein itself, the protein parts of the enzyme. And you see here it's said to be inactive. It's inactive without coenzyme or cofactor. And we see the coenzyme also being depicted as an activator of that enzyme. It's a non-protein part. And we see that when it gets bonded, when you see the coenzyme or the cofactor bound onto the protein part, it forms what you refer to as a hollow enzyme. That is the active form of the enzyme. The active form of the enzyme. So now the enzyme is active and it can have interaction with the substrate, which is depicted red. Now, we saw that cofactors were inorganic metal ions. So here you are seeing some cofactors. So you see copper ions can be cofactor to, say, an enzyme, cytochrome oxidase. Ion 2, ion 3 ions can also be cofactors. Potassium ions, magnesium ions, manganese ions, molybdenum, nickel ions. We saw this enzyme urease. We even saw pyruvate kinase. So you see glucose 6-phosphatase, hexokinase, all these enzymes that some of them we're talking about, you see that they have some metal ions, selenium, zinc ions as their cofactors. You know, what it is that in the structure of these proteins, you have these metal ions that around them you have the protein forming, like they are required for the functional structure of the protein. If they are not there, the protein will not have that functional structure. So the protein cannot be active and carry out its activity. Not only in enzymes, you see, even for instance, if you take hemoglobin, which is or myoglobin, which are involved in the transport of oxygen, you see that you have ion 2 that is at the center of the protein molecule. And many proteins are like that. They have some metal ions. These are the metal ions that we usually refer to as minerals. So sometimes when you go to buy some multivitamins, you see you also have some metal ions being written there as some macro and micro elements, or just minerals, in addition to the vitamins that are on the label. Then the coenzymes. These are also non-protein, the non-protein part of the enzyme, but they are organic. And I mentioned to you earlier that these are some derivatives of vitamin Bs. So we see in the first column, you have the coenzymes. The second column, you see the activity that they are involved in, like the type of reaction that they are involved in. And in the third column, we see the vitamins from which those coenzymes are gotten. So actually the vitamins are like inactive forms of the coenzyme. So when you consume the vitamin gets activated to the coenzyme. Some is just by addition of some phosphate group on it, and then it becomes active. So we see biocytin involved in the transfer of some carboxylic acid groups, and the precursor or the vitamin from which is gotten is biotin, one of the vitamin Bs. Coenzyme A, you see coenzyme A a lot of times in many reactions, also involved in the transfer of some acyl groups. It's coming also from one of the vitamin Bs, referred to as pantothenic acid. So all of them, deoxyadenosylcobalamin is coming from coenzyme. Is really referred to as coenzyme B12. It's coming from vitamin B12, which is involved in transfer of protons, so as hydrogen atoms, and also some alkyl groups. You see some reactions, you see FAD written on the arrows. That is flavin adenine dinucleotide. So that dinucleotide is coming from riboflavin, which is also another vitamin B, and it's involved in the transfer of electrons. So quickly, it tells you that this is involved in oxido reduct. Is involved as a coenzyme to oxidoreductases. So it is involved in oxidation reduction reactions. Same way we see lipids also involved in transfer of electrons and acyl groups. Another dinucleotide, nicotinamide adenine dinucleotide. You also see it in reactions like they just written NAD. Sometimes you see NADH, NADPH. These are also involved in transfer of hydride ions. That is also another coenzyme involved in oxidation reduction reactions, also coming from nicotinic acid, niacin. Pyridoxal phosphate as a coenzyme involved in the transfer of amino groups. You remember we just saw the reaction where you are seeing aspartate transaminase. So pyridoxal phosphate will be acting as a coenzyme to that enzyme because that's transfer of amino groups. And you see the vitamin from which is coming from is pyridoxine. That's vitamin B6. So you see, look at the name. The name is pyridoxine. That's a vitamin. And the coenzyme is pyridoxal phosphate. So it tells you that pyridoxine only gets phosphorylated. Phosphate group attaches to pyridoxine to make it active as a coenzyme. We see tetrahydrofolate involved in transfer of one carbon groups like methyl groups, aldehyde groups, it can be some ethylene groups, methylene groups, and it's coming from the vitamin. Is here folate is actually what you commonly refer to as folic acid. Folic acid, which is involved in a lot of cell proliferation reactions. It's required in development of the fetus, which is why you see folic acid is prescribed a lot for pregnant women, because it's involved in the development of the central nervous system, particularly the brain of the fetus that is growing in the uterus of pregnant women. So that is required for proper development of the fetus. The lack of it may result in maybe children with some mental retardation, some maldevelopment of the central nervous system. You have thymine pyrophosphate also coming from a vitamin B1, thymine, involved in the transfer of aldehyde groups. Again, here you see thymine as the vitamin, and the coenzyme is thymine pyrophosphate. In place of pyro, you can put di, like thymine pyrophosphate can also be said to be thymine diphosphate. So two phosphate groups. So you see, just addition of two phosphate groups to thymine, and it becomes active as a coenzyme. All right, so now we dive in quickly to look at factors that can affect enzyme activity. The first one that we are looking at is the concentration of the enzyme itself. And here we see that the rate of reaction is directly proportional to the concentration of the enzyme. What it means is that as the concentration of an enzyme increases, once there is substrate there and you have enzyme concentration increasing, it means that there will be more enzyme molecules to act on more molecules of a substrate. And so you have the rate of the reaction, the speed of the reaction will continue to increase with increasing enzyme concentration. That's what it means. The second is substrate concentration. So the first one we're looking at is concentration of the enzyme. Now we are looking at concentration of the substrate. That is the compound on which the enzyme is acting. And here we see that there is a saturation curve. What it means is that there is an increase in the rate of the reaction up to a certain point, and then you don't see any significant change again. The graph just flattens up, becomes constant. The rate of the reaction becomes constant at a certain concentration of the substrate. Why that is that, you see, when you have enzymes that are acting on the substrates and you keep increasing the concentration of the substrate, it means that you are having molecules of the substrate that will be occupying molecules of the enzyme. And it will get to a point that the enzymes will be saturated. All of them are going to be occupied with substrate molecules that they will be acting on. That will be the maximum rate of the reaction, the maximum rate of the reaction that you can reach. When you continue to add more substrates, it will not bring about any change because there are no more enzyme molecules to be acting on extra substrate molecules. An enzyme molecule can only become free to act on a substrate molecule when it finishes acting on a molecule that is bonded onto it already. So you have such substrate concentration effect. What you see here is a saturation curve that you are seeing. You have temperature. So you see, like you say, well, our physiological temperature at 37 degrees Celsius. That is when you have enzymes, acting at their optimum. So that is the maximum rate of the reaction. Now, if temperature is lowered, as the temperature goes down, the rate of the reaction reduces. This is actually what is used in the technology of, say, refrigeration or freezing for the preservation of food. So that if you have microorganisms that act on food, they are feeding on the food and they are releasing toxins into the food. That's what makes the food get spoiled. That process of them, metabolizing the food and releasing toxins is being carried out by enzymes. That's enzymes of the microorganism. So if you lower the temperature at which you are keeping the food, it means the microorganisms that are present will become less active. Their enzymes will become less active. And so you can keep the food for a longer time. Now, but when the temperature increases, you see when the temperature increases above 37 degrees, you also see that the rate of the reaction is lowered. That's because we talk about the fact that you have denaturing of proteins. So enzymes that will be present at such high temperatures will get broken down. And once they are broken down, the reaction will not proceed. So the rate of the reaction reduces. So that's the effect of temperature. A similar effect is seen with pH. But for pH is particularly seen as, we have different enzymes that are active at different pH conditions. So for instance, if we have in our mouth pH condition, which is slightly just hovering around neutral pH, it means that if you take an amylase, which is active in a maybe slightly basic and neutral pH, and it's gone to an acidic medium, like in the stomach, that enzyme becomes inactive. It's the same way if you bring an enzyme like pepsin that will be active in an acidic medium to a basic medium, it also becomes inactive. So every enzyme will have some optimum pH at which it is operating, such that when that pH condition is altered, the activity of the enzyme will also reduce. So this is where we bring our class to a close, and not a close, I mean, I've finished talking, and so I'll open the floor now. If you have any question, any contribution, any concerns,